To be a very multi-purpose microscope. So it can do confocal imaging, but it's also set up for doing wide field fluorescence and classical bright field imaging. So it can handle most types of samples. And we'll show you sort of the basics of how to do all of those types of imaging. There's also a lot of sort of more advanced specialized things that it can do that, for the sake of time and not totally overloading you, we won't like go all the way through today, but I'll try to show you like at least what the capabilities are so that you know that they're options, and then if you want to use them in the future, then we can do more specialized training. Alright, so starting off, we'll just sort of go through what the hardware is and how to turn everything on and get it going, and then we'll start with the actual like, image acquisition. So everything is run off of this computer here. Everybody who's gone through the training then will get an individual user account, so that's something we'll do for all of you guys today. Alright, and so when you log in, this is pretty much what your desktop will look like. There's only one software program that you need, which is this one, LASX, which is the Microscope software. Just so I know where we're working from, have people done confocal imaging in the past?
I have not used confocal.
I did like fluorescence.
Yeah, me also.
Like classic, like wide field fluorescence. Yeah. Okay, cool. So some people, I mean, have started with like no microscope experience, so no problem. So then, sort of moving around this way, all of this here is for doing live cell imaging. So we have this, which is this little live cell chamber, which can fit on the microscope stage. And this allows you to control temperature, humidity, CO2, and oxygen levels. So it's basically like a little tissue culture incubator. It all runs off of this touch screen here, which turns on with just a single button on the side. And this all runs separately from the rest of the microscope. And so it'll warm up in a few seconds. Then you can hear it starting to draw in air from the room here. And we've got our temperature, CO2, oxygen, and humidity readings here. And then it's showing you what the set point is and then what the current value is. And you can just click on them and change the set point to whatever you want. Say, for example, you were doing a hypoxia experiment, then you could, instead of being 20% oxygen, you can reduce this all the way down to 1% oxygen. And then once it gets going, you'll see it'll have like graphs, and you usually just give it a couple minutes to everything to stabilize before you put your cells in. And it's run off of gas cylinders here, plus the room air. It can handle a variety of different types of samples. So if you take off this little heated lid, there are different inserts which can go into this to hold different sample types. So for example, this one can hold any sort of standard multi-well tissue culture plate, versus if you had samples that were in tissue culture dishes or chamber slides, there's a different insert, and they just magnetically click down in there, and then this whole thing can go on there. And they rate it as being good for up to like 24-hour experiments, although usually people haven't gone longer than like overnight time lapses. Alright, so that's there if you want it. Now to turn on the actual microscope itself, we start with this down here, which is the power supply. To turn it on, it's these two switches and this key. The light bulb in one of the switches is dead, but it still works, so don't worry that it's not lighting up. Then the second thing to turn on is this here. This is the basically interface between the microscope and the computer, and it's just a single switch in the front there. And when we turn that, we'll see that the microscope starts to turn on. And then the last thing, which is optional depending on what type of imaging you're doing, is this here, which is the metal halide light for wide field fluorescence. So if you were just doing like bright field like histology, you can leave that off and not wear out the bulb in there. But since we're going to be doing fluorescence imaging, we'll turn that on. And again, it's just a single switch in the front. And you don't need to adjust anything on here. That's why I've taped over it, because people have messed with it.
Just excuse me. So you started first this one, then...
yeah, so basically we just work our way upwards. So just start at the bottom.
That one.
This one, number 1, number 2, and number 3.
Okay.
And then when we turn it off, it's just the exact opposite.
Okay.
All right, so now the microscope has woken up. And so you can see we've got this touchscreen panel on the front. And then there's little tabs on the left-hand side to go to different menus. So this first one with the picture of the microscope, that's just telling us like what the current configuration of the microscope is. Then this next one with like the little color wheel, this is to choose what imaging mode that we're in. And so as I mentioned, it's set up to do lots of different types of imaging. So under transmitted, here this is for transmitted light imaging, you see there's 4 options. So BF is your classic, just standard bright field, it's just going to shine a white light through your sample. pH is for phase contrast, so that'll give you better images of unstained cells, for example. And half of the objectives on the microscope are set up with the phase contrast rings to do that. Then DIC is another contrast technique for unstained cells, which generally gives you actually a better image than phase contrast, but it doesn't work if your cells are on plastic. They have to be on glass. And the other half of the objectives are set up for doing that type of imaging. And then the last is polarized light, which can be used for some specialty things like some fibrosis stains, for example. Then under incident light here, so there's just one option, fluo. This is wide field fluorescence. And if we click on that, then we can choose our filter cubes, 4 filter cubes, DAPI, GFP, Texas Red, and Sci5. So those correspond to your standard like blue, green, red, and far red fluorescent dyes. And you can see we've got a shutter control here, so if it's gray, that means no light is being sent to the sample. And if we click on that and that's yellow, now we can see light is being sent to the sample.
Colin, just a question. So I'm familiar with these 3. How can you tell a little bit about this one?
So this one, Wi-Fi, so Sci5. So this is further to the red end of the spectrum. Okay. So this you actually will not be able to see by eye. It's beyond the human visible spectra, but the camera can detect it.
Okay.
And we'll see in the software, it'll show you the filter cubes and you can see the exact wavelength ranges for all of the filter cubes. Oh, and then this guy here, this is our stage controller. So you can see here we're sort of like off the sample. So these knobs in the front are our X and Y controls. So now we can sort of position our sample centered over the objective. And then in back, this is our Z control. And the buttons on the side are to switch between coarse adjustment and fine adjustment of the stage. So you can put on X, Y fast, quickly move to our sample. And then when we want to make smaller movements, click precise, and then it will move much more slowly. And same on the other side, coarser, fine, Z adjustment. And then CS, you can sort of ignore these, but CS, this is confocal scanning.
Okay.
And everything that we're adjusting here, you'll see is also adjustable in the software itself. So usually when you're actually like doing your imaging, you'll basically be running everything off of the computer and not really touching the microscope at all.
Okay.
Then the next tab down, this is the list of objectives. So there's a total of 6 objectives on here. And there's basically 2 sets of 3. So the first 3 objectives, this 10, 20, and 63x, these are the really high quality ones that are designed for confocal imaging. And these are also the ones that are set up with the prisms for doing DIC bright field imaging. Then the second 3, 5, 10, 20x, these are lower quality non-confocal grade objectives, but work fine for bright field or wide field fluorescence. And these ones are set up with the phase contrast rings. For example, if your cells are growing on plastic. And the other difference is that they have a longer working distance, which is the distance between the tip of the objective and the maximum distance it can focus to. The confocal objectives have a very short working distance, so the tip of the objective has to be very close to the focal plane of the sample, which doesn't work well, again, if your samples are, for example, in a multi-well plate, which tends to have a very thick bottom. The higher magnification confocal objectives actually can't focus through all the way through that. If you try, you'll just hit the bottom of the plate before you reach focus. So that's when you might want to use this second set of 3 objectives. But usually the first 3, the confocal grade ones, since they're the sort of higher quality objectives, are the ones that you'll generally want to be using if you can. And then this little pair of icons with the eyeball and the camera, that's telling the microscope where to send the light coming from the sample. So if it's on the eyeball, it'll send it up here to the eyepieces. If it's on the camera, it'll be sending it to whatever detector you're using. So if you're in confocal mode, the light will come over here to the confocal detectors. And then if you're in bright field or wide field fluorescence, the light will come over here to the camera. And so this camera is a color camera, which can operate in either color or black and white mode. So if you're doing histology slides like H&E or trichrome, you would put it into color mode. And then if you're doing fluorescence, you would put it into black and white mode. And then the last tab here is just showing you where you are in the Z travel range. So you can see we're all the way down at the bottom here. And then as we adjust the focus, the objectives are coming up closer to the sample. And this can just be helpful when you're like first finding focus, just make sure you're at the bottom, and then usually I just work my way up from the bottom until I eventually reach focus. And one last thing to note on the objectives, is you can see the 63 has this little teardrop icon here. That's to indicate that it's an immersion objective, which means you'll actually have fluid contacting between the tip of the objective and the sample. In this case, it's a glycerol immersion media. And normally, for all the other objectives, you'll see they say dry, and you'll just click on the objective and it'll immediately switch to whichever one. If you're going from any of the dry objectives to the immersion objective, we click on it and it will blink, but you hear it didn't actually move. That's just to give you a reminder that you actually need to put on the immersion media. And then once you do that and click it again, then it will actually move. And it'll do the reverse when we're moving from the immersion objective to any of the dry objectives, to give you a reminder that you need to clean off the immersion media from your sample and from the objective. And we've got lens paper and lens cleaning fluid here. All right, and so other things about the hardware itself. So it's on this floating table to isolate it from vibrations. Particularly important if you're doing confocal imaging, and particularly at like high magnification where it's sensitive to very small changes. So just before you start your imaging, make sure that it's floating. It's run off of this nitrogen cylinder here, and every few months that will run out and we need to swap in a new one. So if you notice that it's not floating, just let me know and we'll swap in a new one. Then, for actually putting your samples on the microscope, so this condenser arm can lean back to give you better access to the stage. And then there are different inserts which go in the stage to hold different types of samples. So this guy is sort of the standard multi-purpose specimen holder. So it can either hold a single slide, or it can hold a 60 or 35 millimeter tissue culture dish. And to put it in, you've got a red dot here, you've got a red dot on the corner of the stage here, and there's little springs in this corner. So what you want to do is just sort of push it in against those springs and down, and make sure it clicks down and is nice and firmly seated in there. Because sometimes when people get me and they're having problems that, oh, my focus keeps drifting, it's because their specimen holder isn't in there properly. It's sort of like sitting like this and wobbling around.
And when you take out?
So to take out, all you do is just, so it's clicked in there, just grab it and pull it right out. And then we've also got this, which holds standard multi-well tissue culture plates. And again, you just sort of push it in against the springs in the bottom left corner. And then just make sure that it's nice and solid in there, and then you can put your plate in. And then this, which is the size of a multi-well tissue culture plate, will fit in there, is actually a 4-slide holder. So you can see there's sort of little clip springs or plastic springs, and you just clip your slides into that, and then you can have 4 slides on here at a time instead of just one.
So slides should be inverted.
Yes, that is a very important thing, and that is something that sometimes people get wrong. It's an inverted microscope, so yeah, you want the cover slip facing down, which is particularly important with those confocal objectives which have the short working distance, because if the slide's the other way up, the higher magnification objectives won't be able to focus all the way through the slide, because they're designed to just be focusing through a very thin cover slip. Here. And let's go ahead and grab samples. Yeah, so you would take your sample we'll start with. These are some part sections, and yeah, cover slip. Yeah. And then just make sure your sample is sitting in there and feels sort of solid. One thing that can happen if you notice your sample isn't sitting very well is people often get like mounting media will like build up around here. I just scraped it off earlier this morning, but that can cause like bumps here, which cause your sample to sort of like sit tilted. So just check before you put your sample on. And then if there is buildup, you can just take a razor blade and just scrape it off. Alright, so now that we've got the hardware on, we can go ahead and start the software. And there's going to be 2 times when we start the software where it'll ask us to make some choices. And so we'll just go through those as they happen. Alright, so this is the first menu that'll come up. And the first choice here is configuration. And you can see it defaults to this machine option. And that's what you want to choose if you actually want to use the microscope to acquire new images. And if we have that on, it'll make sure during the startup that the computer can actually talk to all of the hardware components of the microscope. And so that means that the microscope has to be on before you start the software. If you just want to start the software and work with like previously acquired images, but not take any new images, you can, instead of machine, you can choose any of these simulator options. And that will skip the program making sure it can talk to all of the hardware. So it'll save a couple minutes actually in the startup process, but then you won't be able to take new images. Since we want to take images, we'll put it on machine. And then this one, which says microscope, there's really no reason for there to be any choice. What you always want is just the Dynamite 8, which is the model of the microscope stand. If you ever forget, it's printed right on the side here. And then we've got this option which is resonant, and it's just an on-off option. So this is referring to specifically in confocal mode, what set of mirrors it uses to sweep the laser beams across your sample. So we have this off. If it's off, it's gonna use the standard set of mirrors which come with the system, which can be adjusted in speed. So you can scan more slowly for better image quality, or you can scan more quickly so that it doesn't take as long. If you turn this on, it uses a separate $80,000 set of mirrors, which works like a tuning fork and just sweeps vibrates at one very fast frequency. So you can image extremely quickly, but you can only image at that very quick speed, and you can't go more slowly. So this is used if you're trying to look at sort of very fast live cell processes, like if you're looking at cardiomyocyte contractility or cardiomyocyte calcium flux, where you need very high frame rates. So if you're doing like 50 or 100 or 200 images a second, but you wouldn't want to use that if you're not having to look at something very fast, because it doesn't give you the option of going slower for better quality. And this last one, load settings at startup, you can just ignore and leave that off. So we'll hit OK, and now it's going to go through the process of making sure that the computer can talk to all of the microscope components. And this is a little bit slow.
Quite a process, I believe, yeah. And fluorescence, normal one, simple one, we just put slides, take image, and go home.
Yeah, it is a process, but it does wind up very capable and lets you do some cool things. So this is the second option which comes up during the startup, and it's asking us, do we want to initialize the stage? And so if we say yes, the stage is going to travel around to sort of its limits of travel, and it basically teaches itself where it is, and it needs to go through this little process to use any of the microscope functions where it can come back to a specific location. Whether if that's where you want to mark individual fields, or if you want to define a whole region and have it do a tile scan where it takes overlapping fields and then stitches those all together. And that's a choice where you have to make it in the startup. If you had said no, then once we were in the startup, once we were in the software, we wouldn't be able to use those functions, and you can't initialize it in the software. You would have to completely shut it down and restart it. So I recommend just going with yes. Because as you can see, that only took a few seconds, whereas the whole startup takes a couple minutes. Okay, and so this is what the software will look like when it starts. By default, it will start in confocal mode. For doing wide field or bright field imaging with the camera, to switch between the two, it's just this little pair of icons here. The little picture of a spectra is for confocal mode, and the little picture of what Leica cameras used to look like is for camera mode. If the icons are too small for you, you can use this little slider to change the size of all of the buttons. And then this up here is where we choose sort of what software module we're using. By default, it'll be this TCS SP8, and this is the core software. Then if you click on that, there are these other options. These are all specialized, like, extra add-on packages that we bought for particular imaging types. So for live data mode, this is for setting up complicated time-lapse experiments, where you want to do something more complicated than just taking the same type of image at a fixed interval. This lets you set up like basically any sort of time-lapse. Then the ones which say FRAP, these are for photo bleaching experiments. So that's fluorescence recovery after photo bleaching. Then the ones which say FRET, these are for doing FRET co-localization imaging. And then the one which says matrix screener, this is high content screening software. And so we won't actually go into these today, but just to let you know that they're all available. So we're actually going to start off in camera mode, so we'll just click on that. and you can hear it switched over, and now we're in camera mode. You can see the general layout of the software is the same in both modes. Your image will always be on the right here. Your sort of setting up your channels will be in the middle, and then on the left-hand side, you actually have two tabs. Acquisition, which is where you set up all the details of the image acquisition, and then projects, which will be where your files are. So if we click over onto open projects, we can see at the moment there's nothing there. So we can go ahead and create a new project for ourselves. By default, it just gives it a generic name. We can right-click on it and rename whatever we want. And then we want to tell it where on the computer to save our project. So again, we can right-click on it and go Save As. And then if we go to my computer, you'll see that there's actually two hard drives on the computer. We have this small solid-state drive, which is just for the software. And then we have this 10 terabyte hard drive called Data Drive, which is where all your files should be saved. And if we click on that, we can see that each of the labs has their own files or folders. And so we can, for example, go to the DOS lab, and you can create your own folders here and save your data there. And it's just important to save the data to the data drive rather than the C drive, because the C drive is so small that if you're saving images there, it'll fill up almost instantly and create problems. And also make sure not to save data to the desktop, because things saved to the desktop wind up on this drive. And then once we start taking images, they'll all show up in a list down here under this project. So we'll start by doing bright field imaging. So the first thing we want to do is check that our camera is in color mode. So you can see there's just a toggle between color and black and white right here. So we're already in color. And then we need to tell it that what we want to do is bright field imaging. So under here where we set up our channels, so at the moment we can see it's saying fluo, so it's trying to do fluorescence imaging. So we just click on that and say we want to do transmitted light bright field imaging. And now it'll say that, and we can see it switch to the bright field filter cube. And then to adjust our exposure, we've got our exposure time and our gain on the camera right here. And then we've got our illumination intensity here. So to start seeing our image, it's always this live button down here in the bottom left corner. So we can click live. You can see we don't really see anything, because probably one, our exposure is low, and two, we're probably very out of focus. Since we had started, if we see all the way down here at the bottom, so usually you can focus by looking at the screen. I tend to do it by eye, at least to start. So I'll just put it on the eyepieces, and now I can. First, we need to go to where the heart section is. Okay. Stop and live again. Okay, and now we can see our tissue. So we can see maybe we want to bring up the exposure a bit. And then adjust our focus.
Wow.
And so this is a trichrome stain, so just healthy muscle tissue is pink, and then fibrosis is blue. For example, over here. If we're doing something like this trichrome staining, which is color critical, particularly if we're going to be trying to quantify it based on color, one thing that we probably want to do is make sure that the white balance is correct, so that our colors are accurate and they're consistent between every time that we image. And so what we can do is, if we go to a blank white area of the slide, and then sort of defocus. Also, I want to check, somebody may have messed up the condenser. Oh no, it's okay.
So how did you go to white area? Just scrolling around? Okay.
Yeah, just using the stage controller and just maneuvering around to a white area.
Okay.
What we can do is if we click on this button here, it says histogram, that'll then show us our intensities in all the colors. We can see they're all sort of maxed out at the moment, so we can bring down the exposure time. And what we can see, if this was really showing up as pure white, then these blue, green, and red should all line up with each other. We can see that they're not. We can see we've got more blue, and so we've got this blue tinge to the image. What we can do to correct that is this little blue, green, red button here, which has color controls. If we click on that and click on white balance, then it will go ahead and take like a reference image, and basically we're telling it this should be pure white, and then it will adjust the blue, green, red intensities to make this pure white, and then use that for all these subsequent images. So you can see these maxima all line up now. So this is our reference for a pure white.
So this is like a compensation layer.
Yeah. And that's just to make sure our colors are accurate. If you're doing something where it's not sensitive to colors being perfect, then you could skip that. But
so do this process is required for all type of imaging, or
this is just for bright field imaging.
Okay. And especially for the quantification purpose.
Yeah, where it's, for example, if you're trying to find like what % fibrosis is your heart section in a mouse which has had some sort of injury, for example, and you're going to be using software to determine how much of the area is healthy tissue versus how much is fibrosis based on extracting the red area and the blue area, you want those colors in your images to be accurate then. And you want it to be consistent between all your samples so that basically you can create one set of settings and then apply that to your whole batch of images and get consistent results for all the images. And then the other thing, which is sort of compensation, is compensating for the potential that the illumination intensity isn't perfectly even across the whole field of view, and that sometimes it's a little fainter, for example, in the corners of the image than in the center, which doesn't tend to be a problem if you're just taking a single field, but if you're doing a tile scan where you're taking lots of fields and then stitching them together, then that sort of dimness on the edges can make it obvious that it's multiple fields put together instead of looking like one nice image. And so there is a tool to help with that, which is this shading correction tool. And so if we click on that, it creates this new little wizard for us. And so we'll go here and it walks us through all the steps. But basically what we do is we click this store position, so that'll just save where we are now so we can automatically come back to it. And then as we did with the white balance, we'll just go to a blank area of the slide and then defocus. And so now this will be what our reference is. And then we go to acquire, and then it can either take a reference image for all the objectives or just the objective that they're using. I suggest just doing the one that you're using. And so we'll click single reference here. And now it's actually going to take multiple reference images, and what it's doing them is it's taking them at different image formats. So you can see these, what it's calling binnings. So what this, what went on there? What this is the camera can basically combine multiple pixels together into sort of like super pixels, which gives you lower resolution but higher sensitivity, which is, you don't usually need to use that, but for example, if you had a very weak fluorescent signal that you were struggling to detect, then you could use that. Alright, so now it's gone ahead and done that. So now we can go to restore and restore XYZ position, and then it's just take us back to where we started from. And now what we can do is we actually have to then tell it we actually want to use that reference, so we'll activate that.
Excuse me. That restore part I just missed. Where did you go for restore?
So it was the last step on the wizard, and it just takes us back to the same position and focus that we had saved as the first step. It just makes it easy to just get back to where you started because you had to move off the sample. But you can always just manually move back to the sample as well. And here, so we'll tell it to activate that. And then you can either do it just on the acquired images or also on the live images. I usually turn off it for the live images and just have it on the acquired images. But you can sort of see if we turn this on and off, that sort of, if it's off, so there's no shading correction, and then you turn it on, you can see it's brightening up the sort of corners of the images. And then if we have our reference image, this turned from gray to green, saying that we have our reference image to use now. If this turns red, that means you've changed some things, for example, like switching to a different objective that you didn't have a reference for that makes your current reference incorrect. And so then you would want to take a new reference or turn off the shading correction. Because if you try to use the incorrect reference, your pictures are going to look really weird. And so if we just want to take a single image, all we have to do is click Start. And it will have captured our image. And now if we go under Open Projects, we've got our file. And again, it just gives it a generic name, and you can right-click or F2 and rename it whatever you want. And now say, instead of just taking a single image, what about if we want to image multiple fields or image the entire tissue section? So they have a nice feature called Navigator, which basically lets you take sort of overview images and then use the overview images to define the individual fields or regions that then you want to acquire. And it can be in a totally different mode, the overview image versus the actual acquisition. So you could do wide field fluorescence with a low magnification objectives to quickly do an overview of your whole tissue section. And then you could pick individual fields and say, go back to these specific fields, but do it in confocal imaging with a higher magnification and higher image quality, or do an entire Z-stack at these positions. And that's particularly helpful if you're saying, I want images in a certain region of the heart, if I want a certain number of images in an infarct border zone versus in a remote region. It helps to have that. Or if you're looking for rare events, so you want to do a quick scan of the whole heart, find your rare positive cells, and then have it come back and do confocal Z-stacks at those specific interesting locations. And so the way it works, if we go live here, we can see we're still in the same position. If we click spiral, what it's going to do is it's going to go out in a spiral from where we currently are and build up this quick sort of overview image. And we could either do that with this objective that we're planning on using, or to make it quicker, what we can do is switch to a lower magnification objective. We still have all the same image controls, but they're now in these tabs on the left. So you can see here, this is where we set up our channels. Here's where we have all of our sort of camera parameters. And so we could say switch to the 5x objective. And so now we can see our field of view is substantially larger. Click live, and then we're hit spiral again.
So for tile scanning, it's better to go for 5x.
So this is just to find the whole tissue area and set up our tile scan. Then we can tell it to come back and actually do the tile scan with whatever objective we wanted. Okay. So now that we're able to see our whole tissue section, and if you have multiple sections on a slide, you can go and do this for each of your sections. Now we can define either individual fields that we want. So we could say, oh, I want a field there, and a field there, and a field there. And these will show up in this task list down here. And then it would just go to those particular fields and image them. Or if we want to do a tile scan, we've got these shape tools down here to define the shape of the region that we want. So a circle, an oval, a rectangle, a polygon. So we can just sort of draw a rectangle around this guy, or if you had a very like irregularly shaped section, say like a long strip of tissue or something, or your section is sort of like got cut weird, then the polygon tool is helpful, and you just sort of click around the region that you want. And again, if you have multiple slides or multiple sections on a slide, you can set up multiple different regions, and they'll all show up as different tasks in this task list. And now, say we've used the 5x objective to like find and define our region, and with the 5x objective, it's telling us it would take a 3x4 grid of images, and so that's what it's telling the tiles here is the total number of fields. But say we actually want to do it with a higher magnification objective, all we have to do is just click on which objective we want to use,